Welcome to our last session of the second day of PyData Global 2021. My name is Yana. I'll be your host today, and it's such an honor to be here hosting Dr. Sandhya, right?
That's right.
Yes, Sandhya, in this session. So, her research, actually, it's before that. Dr. Sandhya is a research scientist at the Moffitt Cancer Centre in Florida. Her PhD in computer science is from University of Basel, and her master in intelligence systems is from University of Edinburgh. Her research deals with developing statistical theory and Bayesian inference models, particularly to problems in cancer biology and computer vision. Prior to academics, she was an assembly programmer working with mainframe operating systems at IBM Software Laboratories and has developed mainframe applications. She has completed 4 out of the 6 World Marathon Majors. Impressive. Today, she's going to talk us about Sparcle assigning transcripts to cells in multiplexed images. So the connection is yours.
All right. Hello, everybody, for being here. And thank you very much for the opportunity for me to present my work here. I'm Sandhya Prabhakaran, and the title of my talk would be Spatial Reassignment of Spots to Cells Using Maximum Likelihood Estimation, which in short we would call a Sparcle. This was work that I did along with Tal Navi and Dana Pear while I was a research scientist at Memorial Sloan Kettering Cancer Centre. And currently I'm at the Department of Mathematics and Oncology, Moffitt Cancer Centre, Florida. So the roadmap for today would be what are spatial transcriptomics, what are the images, what is single-cell RNA-seq data, and what are the challenges we find there? What has Sparcle contributed? What was the reason we developed Sparcle? What were the assumptions and the computational workflow? And a few results and the discussion towards the end. So feel free to ask questions. If I see them, I can field them. Otherwise, I can take the questions in the end. All right. So spatial transcriptomic imaging, one example is that of larfish. Let me show very quickly an example of such an image. But what you see here are illuminated regions, if you can see the mouse pointer, where each of this is a cell, and there are spots that are illuminated with a higher frequency, let's say. So every such circle is a cell, and this could be of any particular tissue. What you're seeing here is that of a mouse brain region. And now you're able to actually image subcellular regions or within a cell. So ideally with a microscope, you would have been able to see a cell, but now you can also see objects or spots within the cell. That's the most simplistic example one can give for this. Now, this is what happens when you want to segment the cells, because just by seeing an image, you cannot quantify it. One way to quantify it is by counting the number of cells. So you actually see those red outlines, which are actually the cells that are there in this particular FOV, or a field of view, as it's called. And here I've zoomed a particular section here, as you can see. And now you can see the red outlines are the cells. And every dot there is actually a gene or an mRNA transcript that we call in the computational biology world. And this is an example like of one spot of one particular color is one relating to one gene. So already you can see that there is an issue with the cell segments being drawn. For example, if you look at this region here, you're not even sure if the spots or the mRNA. So throughout the talk, I'll be interchanging the words spots and mRNAs and transcripts. They all mean the same thing. It's just a spot. So you wouldn't know if you have to assign a spot to this cell or to the cell on the left. And this is a big problem. And that also makes the segmentation very challenging. So the number of cells can be just an approximate number and not perfect. And this image is taken from VisGen's data release. I've also given the link there. This is one of the initial data sets that MERFISH, this particular technology, had out there. This is a single FOV or a field of view where you have 80 cells, you have 80 round circular structures and 140 genes. So Each of these genes would actually be represented as a dot, which you don't see in this figure yet, but we come to it. And MURFI stands for multiplexed error-robust fluorescence in situ hybridization. This is just one FOV. For the entire data set, we had around close to 500 FOVs that we had to go through. And each FOV's stats is what I've given here. You had around 4.1 megapixels to actually process. There were 7 Z-planes deep, 44 channels were imaged. And we had to actually filter out specific channels that were actually demarcating the nuclei of the cells, which is how you would know if the cell is present or not. And for them, for this particular data set, we had 2 different nuclear markers that you could use, poly A and DAPI, to identify the cells. And this is again taken from this particular paper. I've again given the link here. I want to also show another example, just like MURFISH. It's known as HiSeq, probabilistic cell typing by in situ sequencing. This is a figure, again, which helps you to see subcellular structures. This image that you see here is taken from the interstitial part of the brain. Every color here is actually a different cell type. In the brain, you will have like neuronal and non-neuronal cells. So you also have like, are they macrophages? Are they astrocytes? Are they excitatory or inhibitory cells? So each of these cell types have a different color. And the part that we're interested in are these blue spots. These are the genes that actually fell outside the cell and weren't actually being able to be assigned to the cells. Because assigning the genes to the cell is what constitutes what cell it is. So if you have a few genes that fall into a cell, you'll probably be cell typing it or categorizing that cell differently or incorrectly. So this is where the code that we developed called Sparcle comes in. But before that, let me just explain some of the important properties and why this is the most, let's say, fashionable thing happening in the combio world in the imaging field. So spatial transcriptomics, as we saw, was designed for assigning cell types based on the mRNA readouts to their locations. And this is also to determine the subcellular localization of mRNA molecules, because sometimes even the location of these mRNA molecules would be would help us identify what that particular cell is going to do. This is now, as I mentioned, is exploding after an initial period of technological incubation. This was recently named by Nature Method as the method of the year 2020. Genome Biology, yet another big journal, has identified spatial transcriptomics as one of the 11 critical grand challenges. And there are large consortia like the Human Cell Atlas, BICCN, the Allen Institute of Brain Science, and the Human Tumor Atlas Network that are undertaking these large data generating initiatives and putting the data out there. So you as a researcher can actually get access to this and try to understand how difficult and what the problems are with these data sets. And like I've shown before, some of the examples are MURFISH and PySeq, but you also have SMFish, StarMap, and some others that are coming up. Now, I want to just switch gears and explain what single-cell RNA-seq data is. I don't know if many of you in the crowd do know it, but let me just give a quick summary about this. Here what we have is we're taking a tissue of interest. Here what's shown is, let's say, a tissue from the brain. It may or may not be tumorous, but let's say it's a tissue from some section of an organ. You isolate the cells that are there by, again, a huge biological protocol. And then you identify what the cells are and what the genes are. And then you're interested in what this count matrix looks like, where every row can be a gene and every column is a set. So now you have this very interesting count matrix, which data scientists would then now start doing fancy machine learning techniques or deep learning techniques like that of clustering or graphical network building, etc. And this is what will tell you what are the different cell types that are present, how these cell types are connected, what are their lineages, like which cell came before the other in terms of time series analysis, etc. So this is again another very growing field. And a few words about the single-cell RNA-seq data. This is again allowing you to identify an entire genome transcriptome-wide, and you are able to identify what the cell type clusters are, how much what are their proportions, and is there anything new or are there any novel hierarchies that you find? You're also able to have a much clearer view of the dynamics of the tissue and the organism development. That's what I had mentioned before about lineage tracing, and on the structures within the cell populations that are so far being seen as homogeneous. This was also highlighted as the method at the end 2013 by Nature Methods. Single-cell measurements, both from the RNA and the DNA level, as well as more epigenetic marks, are now being able to be collected. So again, there are lots of atlasing efforts wherein you actually are able to get large consortiums, collect single-cell RNA-seq data from different organs of both the mouse as well as the human. And now it's again up to the researchers to identify what the different cell types are, how cells grow, how do they start talking between their cell types, what are their cell lineages, etc. So these are huge data atlasing projects, and for spatial transcriptomics imaging, these atlasing efforts are currently underway. All right, so now I'll start with Sparcle, but I wanted to know if there are any questions at this point.
Not so far.
Okay,
Very clear.
All right, that's okay. All right, so let me start with Sparcle, what the research question was. So on panel A, you can see again a similar FOV or a field of view that we saw before with so many cells. And here we actually see that anything that is in green is actually what quantifies the cell. And panel B is the same FOV that we see in panel A, but here you can see in a more zoomed in section that there are blue dots that are actually the mRNAs that are within the cell. So these are segmented and they're within the cell, whereas the pink crosses are those mRNA transcripts that are outside the cell segment. So it almost looks like 50% of the mRNA transcripts are within the cell and 50% are outside. So, and panel C actually shows the same FOV, but where they're colored now. So you can also see how different those mRNA transcripts are and how much of information we actually lose by not capturing those spots or mRNA transcripts that are now outside the cell. So this is where Sparcle comes in. We actually want to recover what we call as dangling transcripts and assign them back to cells. So the assumptions we make here is that we do not say that there is a uniform distribution of spots within a cell, as we saw before. There are lots of different genes within a cell causing heterogeneity. And there's a large number of transcripts in each cell, which is why we can consider this to be as a multivariate Gaussian distribution and do all sorts of multivariate statistics. And we've also checked this using the Lilly-Force test. The inputs, as we are now clear, we need some fish FOV images, imperfect cell segmentations, because we need something to start with. And we also need to know which mRNAs belong to which gene. So these are already provided. And Sparcle will take that in and give you an improved spot to cell assignments. It would give you a more realistic per cell quantification of that gene and a better delineation of the cell boundaries, and also improves the cluster assignment. So I can quickly show. All right, so before going there to the graphical abstract, let me also go through the principles first. How we actually do this is first we generate a count matrix, which is nothing but the cells and the genes that are present in an FOV, which you can then use for clustering. And you can use any clustering algorithm of choice. Next, you construct what's known as a mock cell per dangling transcript. And this is another figure that we've created just to illustrate this. So in the same FOV that was shown before, we can zoom into this particular section, and you can see for an example dangling transcript that was not assigned to the cell, let's say this pink cross, we draw a circle around it to call it as an approximate neighborhood, and you quantify the different crosses of different colors that are there inside that neighborhood and construct what's known as a mock cell. So this is now an ordered vector, which is arranged in the number of genes. And you can actually, it's just a vector with just numbers filled in. Every time you see a brown, you add a 1 to the brown column. And this keeps going on for every such cross. And you can also weight these mock cells based on how far they are from a particular spot, a particular neighboring transcript. And the method we do it is by using a Shepherd's method. And then finally, we use a maximum likelihood estimation for assigning the transcripts to the cell. This is, again, pretty simple, cheap, and doesn't require us to customize this for different protocols. And it's the code has also been parallelized. And then once this happens, you can update that particular cell entry in the global count matrix, cluster, and iterate. And in our observations, we've seen that 3 iterations is enough. This is a graphical abstract for anyone who's interested, and this is like a take-home, where you actually see the FOV that we started with. I zoom into this particular section here, and you can see the different cell types and the different spots and dots that are there. And some of these cells don't even have the cell segments segmented properly. So you see a lot of those dangling mRNAs outside. And here again, we elaborate about how we build a mock cell for each of those dangling transcripts. And eventually you're able to assign each of these crosses to the nearest cell, which has a similar feature. And that's where the maximum likelihood estimation comes in. And then from your initial FOV, you've already had an original baseline genes times cell count matrix. You update that count matrix now with the observations you have here, and you continue this process until you have a much clearer and, let's say, more correct count matrix. And these are just a few examples of how Sparcle goes through these iterations. It's the same panel from A, B to C. Just that this is the first iteration where you see how every black dot is an mRNA that gets assigned to the nearest cell. So is the case with B, the second iteration. And the third iteration, we combine all of these mock cell assignments to the cell. So eventually you get these sparkles, and this is why we've also named it as a sparkle. This is again showing the number of cells that we've recovered for each of these different cell types that's actually present in the brain hypothalamus. So the yellow, so this is now comparing with single cell RNA-seq, MERFISH alone, and MERFISH by using Sparcle. So that's basically the magenta. And we can see that the proportions that we obtain via Sparcle are kind of in close concordance to what the single cell RNA-seq also dictates, because this is like considered as the holy grail, the single cell RNA-seq data. And this is again another visual showing the fraction of the transcripts or the spots that were recovered. And through Sparcle, the pie slices that are in darker pink is what we've recovered. So rather than just retaining the image with just 50% of the FOVs, 50% of the transcripts, you're now able to at least get around 35% back into the cells. And I also show the comparison of Sparcle both before and after usage on the MERFISH data. The point of here is we're actually doing a cross covariance matrix between the different cell types. And you can see that this is before and this is after, where you compare MERFISH data with single cell RNA-seq. And this is the MERFISH data that's applied to Sparcle, and then you compare a single cell. You can see that the diagonal is fairly strong and highly red in color, which means that we were able to find a stronger correlation of tightly connected clusters post-Sparcle. And this is again just showing you the same thing. Post-Sparcle, we are able to extract for a set of genes, which you see as the rows, much brighter correlation between the cell types, which are actually shown in the columns. And I showed the same example, but for SM-FISH, which is yet another spatial transcriptomics imaging technique. The same thing here, you can now see there's a much stronger diagonal, as well as there's a clearing of interactions between cell types, which is something you do not want to see. So we have more clearer diagonals and off diagonals. And here is again, this is probably more biological, but what this does is it says that we are able to clear up many of the interactions between genes that are shown here in the rows to the clusters that were there in SM-FISH. And then finally, this is where we are right now. So I mentioned that we use a maximum likelihood protocol that to assign each marked cell or each dangling transcript to the cell. We'd actually like to change this and add a prior distribution where it's based on the location of the transcripts, and also implement a maximum a posteriori Bayesian inference procedure. Currently, this is written in Python, and as you would know, or as you already noticed, the mark cell creation is kind of computationally intensive. So if you have to blow this up to large number of images, this can get very tedious to run. So another idea, another direction we have is that of coding this up in another language that's faster. And that's also another reason I reached out to this community to know if anyone is interested to discuss this more and see how we can make this faster. And this is another general issue in the field, that we would like to have better cell segmentations that can trigger better clustering and a lot more of fancier methods in this space. The paper is currently here. You'll also find the GitHub link to the code in the paper. These are different ways to reach me. And I'd also like to acknowledge our funding sources. I was partially funded by the Python Software Foundation, and NIH was the funder for the group where I had done this work. And this is all that I have. I'm very happy for your attention, and very happy to take any questions. Also, if you'd like to collaborate, please reach out. Thank you very much.
Thanks you, Sandhya. Thank you. Your talk was super nice and super different from my normal daily basis.
So. Yeah, this is hardcore academic research.
Yeah, no, my background is in physics, but it's like it's far from I was in. I will just maybe give to the attendees like a couple of minutes to write some questions that they can have.
Yeah.
And in the meantime, I have a question. Maybe
should I stop sharing the screen, or is this fine? Should I stop?
It's up to you.
Oh, okay, yeah, you can go ahead. Yes.
Okay. Yeah, at some point you mentioned that we can use any type of clustering method in the Sparcle workflow.
Yes.
And I was thinking that the success of this clustering task depends on the shape of actually the data points that we have. Do you see any pattern in your data points, in your cells? So we can privilege it, one technique over the other one, or
okay. So so I think you have multiple parts to your question. So one of the things that both of these algorithms have in common is that you do not have to fix the number of clusters. So let's say in k-means, you have to fix what k is. But in phenograph, which is very frequently used in clustering biological data sets, as well as the Gaussian mixture model, you do not have to mention the number of clusters. So automatically it would tell you what it is. So I think it is already known that we do not assume the number of clusters to begin with. To also answer if there is a pattern, there is already an underlying pattern in the data, because if you, let me just go to an image, probably here, right? So each of these regions, like you can see there's a band in between, there are those edges, they are already spatially allocated patterns, right? So now what we're doing is we're assigning those spots that are left out to these patterns, right? So now we're strengthening this pattern based on our assignments. So that's the beauty of using images versus just single cell data. So in single cell RNA or single cell ATAC-seq data, what happens is you have access to the entire genome, but you do not know where they're coming from. That's how the data is being processed. But in images, you can actually see where they are. So you are blessed by the spatial location. And now you don't have to worry about, is there a pattern? There is an underlying pattern. Yes, you shouldn't break it. You can actually use that as your baseline. But you don't have that in the single cell data, in the single cell RNA-seq data, ATAC-seq data. So I think that's a brilliant question. You do have a pattern, and that's dictated by the image itself.
Okay, thank you. Thank you very much. We have like 3 more minutes. Yes, I have a question. Sorry, Ellie, I didn't see. I will read for you.
Yeah, sure.
I noticed that the Sparcle output shows some cells with lots of Sparcle branches and some that are very compact. Is that what you expect to see biologically? Are they different types of cells?
So I don't know at which point this was asked, but I'm assuming it was probably here. If this is the case, yes, this is actually kind of what you would see, because once you go into the images and you look at what these particular genes are, they are very close together, right? Just that they were missed out as part of the cell segmentation. So they would definitely bunch together and they would belong to one of the cells. And I actually don't remember which one of these is which, but that's a good question. Let's say this is, for example, an astrocyte, then all of these greens would be astrocytes. And I don't know if, let's say, if the orange ones were were excitatory cells, then yes, all of those crosses would be genes that relate to excitatory cells. Yes. There's definitely a chance to make an error, which is why we have been very careful and cautious in not just so even if you look at this pie chart here, there are some that we leave out because we are not sure. They might be very far away. We just do not know which one to assign to because it's probably just oscillating between 2 different or 3 different cell types. In those cases, we do not assign them. Whenever we assign them, it would be to the ones that we are 100% sure. Yeah.
Okay, thank you very much. I think exactly Ellie was referring to that slide.
Okay, perfect. Yes.
Yeah.
Okay.
Okay, looking at the hour, we are at the end of our session. So
all right.
I would like to thank you very much to you and also Niels for the nice talks. Niels was the previous one.
Yes, I did. Yeah.
Thank you very much. Also to the attendees to be here. And yeah, I will stop the record right now.